Well, welcome back to part 2 of the urinary system lecture. We're going to go ahead and talk about urine formation. So, urine formation involves several different processes. Let me get my pen right here. And it's going to involve basically filtration. That's where you push plasma out of the glomerular capillaries into the glomerular capsule. It's then that filtrate's then going to go into the renal tubule, so it's going to go into the proximal convoluted tubule. So you're going to have within that tube, the proximal convoluted tubule, the descending ascending loop of Henle, and the distal convoluted tubule, you're going to have tubular reabsorption along the way. That reabsorption can come in the form of reabsorbing water or reabsorbing back into the capillaries various kinds of solutes or even various kinds of nutrients as well. So all along that tube, we're going to have some tubular secretion. So some things are going to be put into the tube, into the filtrate. Could be things like hydrogen ions, ammonia, urea, potassium ions, different kinds of things can be done that way. And then finally, we're going to have excretion, where all the collected wastes and water and leftover materials that's not reabsorbed back into the body is going to go through the collecting duct and then into the papillary duct and then into the minor calyx. All right, so I want to talk about each one of those little steps and give you a few highlights about it. So in glomerular filtration, this is where urine formation is going to begin, and the fluid portion of blood is going to be filtered by that little ball of capillaries, and then the other things, the basement membrane and the podocytes, the filtration slits. So so anything that's squeezed out of that glomerulus and goes through the basement membrane and through the filtration slits of the podocytes is going to become the filtrate, okay, that we're finally going to put into the proximal convoluted tubule. All right, well, how do you squeeze stuff out of that little ball of capillaries called the glomerulus? So the main force for moving substances by filtration through the glomerulus is going to be hydrostatic pressure, that is blood pressure. And so there's going to be competing forces. So the main pressure is going to be hydrostatic pressure, but due to the plasma proteins and the solutes that are dissolved in the plasma of the blood, there is an osmotic pressure in the blood that's going to cause water to be reabsorbed from the glomerular capsule back into the capillary. So that pressure is going from the capsule to the capillary, and that's called blood colloid osmotic pressure. Okay, so you know, if hydrostatic pressure is huge and blood colloid osmotic pressure is very little, then there will be some stuff that will be squeezed through the glomerulus. So it's a battle of pressures as to which way the fluids are going to be moving. Okay, and there's another pressure too that I'll describe here in just one second. So just to orient you, we have the afferent arteriole bringing blood into the glomerulus, so it's going into this ball of capillaries. Here we have the efferent arteriole going out, and you can see the yellow cells are the podocytes that are creating those filtration slits. So we have several different pressures. One of the pressures is the blood pressure, the hydrostatic pressure. And you're going to look in different books, and they're going to have different measurements. I'm going to show you a few different measurements, but in this particular graphic, it's 55 millimeters of mercury is the glomerular blood hydrostatic pressure. So there is another pressure, the osmotic, blood colloid osmotic pressure. And so you can see that it's 30 millimeters of mercury, and it's going in the opposite direction than the hydrostatic pressure. So hydrostatic pressure is basically going this way. Blood colloid is going back the other way. It's causing the reabsorption of water back into the capillary. And you can see it's 30 millimeters of mercury. Well, 30 millimeters of mercury is less than 55 millimeters of mercury, so you know, filtrate will be squeezed out. But there's another pressure called capsular hydrostatic pressure. So the fluids that are inside of this capsule, they do develop a pressure, and that pressure can be measured, 15 millimeters of mercury. So when we look at the net filtration rate, we're going to take the hydrostatic pressure from blood. we're going to subtract that from the capsular hydrostatic pressure and from the blood colloid osmotic pressure. So if you look in the little equation here, we have 55 millimeters of mercury minus 15 millimeters of mercury minus 30, and we get a positive number. If you get a positive number, that means that there will be some filtration, so there will be substances coming out of the glomerulus moving into the proximal convoluted tubule. Of course, if you got a negative number, that would mean that more fluid was going back into the glomerular capillaries. Okay, so there's a lot of things that can upset the balance of blood pressure, capsular hydrostatic pressure, and blood colloid osmotic pressure. So those things, you should be somewhat aware of, and we'll talk about a few of those things. Just to show you one more picture here, here we have the afferent arteriole taking blood in, efferent arteriole taking blood out. So we have the little section here of the glomerular capillary and the podocytes blown up for you a little bit in this diagram over here. So you can see this is where blood cells would be inside the lumen or space of the capillary. This is the simple squamous cells, the simple squamous endothelial cells. You can see they're fenestrated so that fluids can move out and perhaps proteins and amino acids and glucose. There is a basement membrane that is selectively permeable, so there are things that can go through that. So your large proteins won't go through that. And then your filtration slit is even more selective. Only super small things can go through the filtration slit. Once it makes its way through the filtration slit, then it's in the capsule and will move towards the proximal convoluted tubule. And I just basically brought this picture to show you that some texts will have different numbers. So here you have 50 and 25 and 15. So net pressure of 10. So different texts will have different numbers. Okay, well, there's many factors that affect the filtration rate. So some things would include the hydrostatic blood pressure. So blood pressure will affect the filtration rate. The pressure inside the capsule, so capsular hydrostatic pressure, and then of course blood colloid osmotic pressure. If you change any of these variables, it changes how much net filtration you're going to have. All right, well, one thing that can affect something like the hydrostatic pressure is the afferent arteriole. So if you have the little smooth muscles around the afferent arteriole, if they constrict in response to sympathetic nervous system stimulation, then your filtration pressure and thus your filtration rate will decline. Okay, so if you stop sending blood into the glomerulus, of course the filtration rate is going to decline because you've lost your pressure, the main thing that's causing filtrate to be squeezed out. When the efferent arteriole is the little smooth muscles around the efferent arteriole, when they constrict, that will actually take and increase the filtration pressure, or the pressure inside the glomerulus, the glomerular capillaries, and that will increase the rate of filtration. So when you have an upset in the balance of your blood colloid osmotic pressure, so for example, let's say it's really super high because you have a lot of salt in it perhaps, then the filtration rate will decrease. Okay, so there will be a great pressure going back towards, from the capsular space back towards the glomerular capillaries. There will be a great pressure because of that high solute concentration in the blood. So you're basically, it's almost like putting salt on a slug. It sucks the water out of the slug. So if you have a high solute concentration in your bloodstream, it's going to pull that water back into the bloodstream. Let me get the ink off this. And you can also have a capsular hydrostatic pressure that's high, and if that happens, then the filtration rate will also decline. So if you average it out, filtration rate is about 125 milliliters per minute, or about 80 liters in 24 hours. Okay, so 80 liters, how do you kind of imagine that? That would be 92 liter bottles of soda. So imagine 92 liter bottles. That's how much filtration is going to occur in 24 hours. Now, you don't urinate out 90 liters of water, because most of what you put into the proximal convoluted tubule, into the renal tubule, most of that's reabsorbed as it goes through the nephron. Okay, so we just talked about filtration. Let's talk about tubular reabsorption. Remember, reabsorption is where you take materials in the tubule and bring them back into the bloodstream. That's tubular reabsorption. Most reabsorption is going to occur in the proximal convoluted tubule, the PCT. That's where those cells contain the microvilli. They contain lots of mitochondria, lots of ATP, lots of endoplasmic reticulum and Golgi apparatus, and therefore they make lots and lots of carrier proteins. Okay, so that's where most reabsorption is going to occur, is in the proximal convoluted tubule, and it's because they have the machinery, both the surface area because of the microvilli and the proteins to do the work of carrying materials across the membrane of that cell. Now, those carrier proteins can be overwhelmed if, let's say, you have a really super high blood sugar because you have diabetes and you haven't taken insulin in any given, in a time period where you can lower your blood sugar level. That high sugar level may overwhelm the protein's ability to reabsorb sugars back into the bloodstream. Therefore, they're going to be found in your urine because the carrier proteins can be overwhelmed and don't have the ability to process all the materials. So the proteins have a limited transport capacity. Excessive amounts of any substance will be excreted into the urine. So glucose, amino acids, those are going to be reabsorbed by active transport. We have to use ATP or energy to bring those substances across. Water is going to be brought back across through osmosis, so that's a passive process, but facilitated by aquaporins in the membranes of the cell. And then, proteins and some other substances will be brought across by a process called penocytosis, and that's where the cell membrane has little invaginations, and these little invaginations pinch off, trapping, water and materials, and they come in a little vesicle and are processed by the cell. This is a really important diagram. I think it's one that you should really take a look at several times. And if you have the PowerPoint slides, you should print this off in a large slide so you can see what's going on. Now the key here is down here. So we have orange arrows represent active transport, where you have to use ATP in order to transport substances, and the purple arrows are representing passive transport. All right, so we have three major sections, or actually 4 major sections of this tubule that we need to really focus on. Let's focus on the proximal convoluted tubule here. So if you notice in this diagram, the proximal convoluted tubule is passively bringing back in bicarbonate. That's an important thing. You want to bring bases back into the body to maintain the proper pH. The body is continually making acids from the breakdown of foods that we eat. It's constantly making carbon dioxide from the breakdown of the foods that we eat. So bicarbonate is a buffering molecule. We want to bring that back in if we can. Salts are really important. Sodium chloride, is important in muscle movement and also in nerves. So you definitely don't want to get rid of all your sodium. So we do bring that back in a passive transport. In an active transport. We do bring back water passively through the process of osmosis. We bring back nutrients such as amino acids and glucose in an active method, and we can even bring in potassium ions. So what do we secrete? So secretion is taking stuff from the bloodstream and putting it into the tubule. So we do secrete hydrogen ions. Now remember, the body's always trying to become acid or acidic, so we want to drop those hydrogen ions back into the filtrate. That also makes your urine slightly acidic, which aids the immune system at fighting bacteria and organisms coming up the urethra or into the urinary bladder. We also drop off ammonia. Ammonia is a waste product from the breakdown of amino acids, and ammonia is a really strong base, and it's a killer of cells. So we have to get rid of ammonia out of the bloodstream. So that's passively taken in to the proximal convoluted tubule. Now remember, this tubule is really long. It's millimeters long, and it's convoluted, so there's a lot of surface area there. And again, you have microvilli, which also increases the surface area of the inner wall of that part. So in the descending loop, so here's our next our next little section right here, the descending loop of Henle, or the descending loop. We basically are going to have simple squamous cells. They're going to have lots of aquaporins, and they're going to reabsorb water. In the ascending loop, we're going to take care of bringing back in salts. And what's kind of cool about this is when you bring in salts, that creates a hypertonic area here, which helps to absorb water. Okay, so when we bring in salts, water follows it. So water always follows salt. So that's kind of a cool little thing. Those things are right next to each other, and one side brings in salt, one side allows water to be reabsorbed. So this is done passively and actively, and there are carrier proteins that allow that to occur. Next part will be the distal convoluted tubule. So we do bring in actively sodium chloride, we bring in passively water, we keep on bringing bicarbonate in, we're getting rid of some potassium here and some extra hydrogen ions as well. So those are actively transported into the tube. Now remember, this is the collecting duct, so this is a separate little thing from the nephron. And in the collecting duct, the cells there will actually bring actively out sodium chloride that aids in the reabsorption of water. You know, these things are real close together. So anytime you bring out salt, water follows it. Sometimes we even bring out some urea to create a hypertonic solution to bring the water out even more. We don't want to urinate out a lot of water. We want to try to reabsorb as much water as we can back into the body. Okay, so definitely want to go back and look at that and maybe even review that. That's really important to know each of the different steps. The proximal convoluted tubule, descending loop, ascending loop, distal convoluted tubule, and then each thing that it's responsible for doing. And then also the collecting duct. Just to reiterate a point I was trying to make earlier, and that is that when the blood plasma, so if you look at the osmolarity, that is the amount of dissolved solutes per liter of filtrate or blood, you can see that the plasma and the filtrate have about the same osmolarity of 300, and that's many osmoles per liter. So if you look at the filtrate as it goes through, it maintains about 300 units. And then as water's pulled out, you can see the osmolarity increases. So the solute concentration increases as more and more water's pulled out. Now that solute concentration decreases because we're pulling salts out. Okay, then it gets to about 100. Doesn't really do too much in a distal convoluted tubule. But once we get into the collecting duct, so the osmolarity increases. It keeps on increasing more and more as salts are pulled out. As water's pulled out in the collecting duct. Okay, so remember, salts are being pulled out here. Water's following the salt, and you can see the changes in osmolarity as it occurs. Okay, so this is just showing you water's being pulled out of the descending loop, salts are being pulled out of the ascending loop. If you pull out water, the osmolarity increases. If you pull out salts, the osmolarity in the filtrate decreases. So as I keep on pulling more water out of the collecting duct, osmolarity increases inside the filtrate. Notice that we do pull some urea out, and we do that helps to draw more water out, but some of that is passively put back into the filtrate. This is kind of a nice diagram. I like it. It's kind of small to see on the screen, I know, but here you have, just to orient you, you have the glomerulus, proximal convoluted tubule, descending loop, ascending loop, distal convoluted tubule, and collecting duct. Let me take the ink off that. Let me move my face. Okay, so if you kind of look at the osmolarity of the plasma of this blood vessel that's going out here is about 300. But as we get down here, we basically have pulled more salt in, so we have about 1200 going over here, and as we pour more water in, it goes back down in number. Okay, so because we pick up salts over here, we can pick up water over here. Water always follows the salt. So it's just kind of a nice way of kind of looking at it, that if we pull out salts here, then over here we're going to pick up water. It's going to draw the water and move it towards the blood vessel. I also like this picture too because it shows you the solute concentration with these little dots. And you can see as water's being drawn out, the solute concentration inside the filtrate gets more and more, and as salts are being drawn out, the solutes in the filtrate are becoming less and less. And then over here we have very little solutes, but as we begin to concentrate our urine, it becomes heavy in solutes. So since urine's heavy in solutes, that's something you don't really necessarily want to drink a lot of, because it's just like drinking salt water, maybe a little less salt in that water, but you don't want to drink a lot of urine because it has a high solute concentration. I thought I'd go ahead and throw in here antidiuretic hormone. So antidiuretic hormone has its influence here mainly at the collecting duct, and what it's going to do is make for more reabsorption of water. So this particular hormone will cause aquaporins to be formed and along the walls of these cells, and those aquaporins will help to bring water, reabsorb more water back into the body. Now, if you have a lot of antidiuretic hormone that's working, that's going to cause your urine volume to be very, very concentrated in solutes, because water is being drawn out, because that's the job of antidiuretic hormone. Now, you secrete antidiuretic hormone by the posterior pituitary gland, and that's in response to the volume of your blood becoming low. Okay, so it's a signal to make more blood volume, to put more water back into your bloodstream. All right, well, there's some waste products that we create: urea and uric acid ammonia. These are big time important things to get out of your body. Urea is a byproduct of amino acid metabolism. So anytime you break down amino acids, I don't know if you remember, but amino acids have an amino in, and they have a carboxyl or carboxylic acid in. So we do have to get rid of that when we break down amino acids. Also, when we break down nucleic acids, that is DNA and RNA, there is a product called an amino product that has to be taken out of the body. Urea is passively reabsorbed by diffusion, but about 50% of it is going to be excreted in the urine. So uric acid is another breakdown product of amino acids and nucleic acids, and ammonia is a product too that is a breakdown product of these materials. Now, your urine composition will, of course, vary from time to time, reflecting on the amounts of water you're bringing into your body and the amounts of solutes that are in your bloodstream and that your kidneys are eliminating to help maintain homeostasis. The great bulk of urine is going to be water, though. So 95% of urine is going to be water, but it's going to contain those breakdown products: urea, uric acid, ammonia, trace amino acids, trace levels of sugar. There will be drugs in it. Anything that any type of drug will be in it. Antibiotics, medications, all different kinds of medications. Human chorionic gonadotropin if you're pregnant. Electrolytes. All of these things will be found in urine, but main bulk of it's going to be water. If you ever go into becoming a person that's a lab, medical technician, you will be doing urinalysis. That'll be something that you'll have to do. So just kind of looking at plasma versus urine. So you can see that it's highly variable in terms of sodium composition. Notice that urine is going to have a lot more potassium ions. It's going to maybe have more chloride ions. It all depends on the circumstances. And a lot less bicarbonate than the plasma of the blood. So we do want to bring that bicarbonate back into the bloodstream to help maintain the pH of the blood. If we look at nutrients and other substances like that, very little sugars are in urine. Very little lipids, very little amino acids, and hardly any proteins at all unless you have some disease state. So if we look at nitrogenous waste, huge amounts of urea, huge amounts of creatine. This is a product that's going to be found in oftentimes in muscle metabolism. Uric acid is very high, and ammonia is very high in urine. Well, this is another reason why you don't want to drink it too, is that it's mainly made of waste products. So, there are a lot of different characteristics of urine. So pH of urine is going to be anywhere from 4.5 to 8, but it averages 6, which is slightly acidic. In regards to osmotic concentration, it has lots of solutes dissolved in it. It's mainly water. You make somewhere in the order of 700 to 2,000 milliliters per day. So that 2,000 milliliters is pretty high. It's 2 liters of water per day. It has a yellow coloration. That's due to the breakdown products of red blood cells. So you do absorb some of the breakdown pigments of red blood cells that's found in bile back into the bloodstream, and it is excreted out as urobilinogen in the bloodstream. So it's typically clear, and that depends on how much water you're drinking. Typically, the odor will vary based on if you're taking vitamins or have disease states, and then the bacterial content will be 0. It should be sterile. So urine should be sterile unless you have a urinary tract infection. Okay, so if you ever do a urinalysis one day or have a urinalysis, they'll check you for all different kinds of things. So they're going to check you for urea. They'll check you for the breakdown of creatine phosphate. They're going to be looking at ammonia, uric acid, urobilinin, which is the breakdown product of red blood cells, and even bilirubin. So these can show disease states that you may be having based on the composition that are found in your urine. So there are normal values, and then there are abnormal values. So also you will see that there will be also normal values of various things like carbohydrates, ketone bodies, lipids, and amino acids. These can show, again, certain disease states if they're in higher numbers than what you want. And of course, all of your electrolytes too are going to have certain concentrations and will show various kinds of problems based on if they're in your urine in high concentrations. So you do have some red blood cells in your urine. So 130,000 red blood cells per day, or 100 per milliliter, is what you would expect to have as a normal value. If you have excessive numbers of those, then you're going to have then there's something wrong with your body. And white blood cells, something in the order of 650,000 per day will be eliminated, 500 per milliliter. So there's a lot of white blood cells because they're actively trying to fight infections that may be in those parts. Okay, so after the urine forms in the nephrons, urine passes from the collecting ducts to the renal papillae. They then go to a minor calyx and then a major calyx, and then into the renal pelvis. There they head towards the ureters, which are tubes that carry urine out of the kidney. Each ureter transports urine from the renal pelvis by rhythmic waves of motion called peristaltic waves. There's also some pressure there, hydrostatic pressure, and gravity pulls it down as well. There's no anatomical valve at the opening of the ureter into the urinary bladder. So if you take a look, the ureter just drops. So that's the urinary bladder there. So there is no sphincter or valve. It just drips right into the urinary bladder. When the urinary bladder fills, it compresses where the ureter comes into it from the kidney. It compresses it so that you don't get backflow into the kidney. The bladder is hollow. It's distensible, and that means it can shrivel or it can grow in size. It is a muscular organ, so it does have smooth muscle tissue. There is transitional epithelium inside that has the ability to expand and then contract smaller. And it has a 7 to 800 milliliter capacity. Urine from the urinary bladder moves to the outside via the urethra. It's a tube that carries urine from the urinary bladder to the outside of the body. Just to look at some of the male anatomy versus female anatomy. So here's the urinary bladder up here in the male. There is a urethra that comes out. Of course, it goes through the penis and then exits out. Females, here's the urinary bladder here. And then the urethra goes out this way, so that urine goes out that way. You can see that pregnant females, as the baby grows inside the uterus right here, it will put great pressure on the urinary bladder and make it smaller, so there will be more frequent urination in a pregnant female, or at least the potential for it. So just showing you the urinary bladder. Here are the ureters. So there's one from the right kidney and one from the left kidney. Here's the orifices where they drop urine into the urinary bladder. Inside the urinary bladder, it does have a kind of a surface feature that's almost similar to the stomach. It has rugae, which is folds, and then of course it smooths out as the urine collects inside the urinary bladder. Notice that there is muscle tissue there, so we have plenty of muscle tissue to contract to send urine to the urethra. Okay, there are sphincters at the outflow of the urinary bladder, so we can control when we go to the bathroom, at least when you're old enough to do that. Okay, and so this is a male urinary bladder. You can see it's going through the prostate, and females don't have that particular gland. Micturition is the name we give urination. This is a scientific name for urination. And this is the discharge of urine, and it involves voluntary and involuntary muscle contractions. So there are stretch receptors that trigger a spinal reflex for urination. We control this or learn to control this in early childhood. The urethra will carry urine from the internal urethral orifice to the external to the exterior of the body. In males, it discharges semen as well as urine, so there are multiple tubes that come together in the urethra. Okay, so that's a little bit about the urinary system. I want to finish up by talking about hormonal regulation of blood volume and blood pressure, and then finalize with focusing on how the urinary system is involved or integrated to every other system of the body. So I'm just going to put my face way over here. All right, this looks like a pretty busy graph, but basically we want to look kind of right here to start it off. So our stimulus for this complex process that you're seeing here is a decreasing blood flow to the kidneys. So we're basically looking at a drop in blood pressure, perhaps a drop in blood volume. This is going to be a stimulus. Now that stimulus is going to be detected because there's a decrease in filtration, there's a decrease in pressure, decrease in urine production. All these things, there are sensors to sense that. One of the effects is going to be the juxtaglomerular complex, which is that little series of cells in this little triangular area right down here, is going to release renin into the bloodstream. Now, renin is going to trigger it is an enzyme, and it's going to basically take angiotensinogen and make it into angiotensinogen 1. There are enzymes in your lungs called angiotensin converting enzymes, or ACE, and they're going to convert angiotensin 1 to angiotensin 2. Angiotensin 2 will have several effects. One of the effects of angiotensin 2 is it constricts the peripheral arterioles, therefore it's going to increase the pressure. So if we follow this line on down here, there's going to be increases in blood pressure because of that, and that may increase the flow of blood to the kidneys. All right, let's get some of this ink off. So angiotensin 2 also is going to cause an increase in aldosterone secretion by the adrenal glands. So the aldosterone is going to increase sodium retention. That sodium retention will increase the amount of blood volume, and if you increase blood volume, it increases blood pressure, thus it goes back to maintaining homeostasis. So, and then there are neural responses too to angiotensin 2. So the brain will sense that level of angiotensin 2 increasing. There's also baroreceptors as well that can sense changes in blood pressure. But this is going to increase your stimulation of thirst centers, which will increase fluid consumption, which will increase blood volume. They're also going to the brain will also release through the posterior pituitary, it will release antidiuretic hormone, which also increases fluid retention, which increases blood volume. And then sympathetic nervous stimulation can happen as well, which can increase cardiac output. And then in addition to all the other things, will cause general vasoconstriction, or the narrowing down of arterioles all throughout the body, which will increase blood pressure. So if we have an increase in blood volume and an increase in blood pressure, this will increase general circulation blood pressure and thus increase the flow of blood to the kidneys. It's very complicated stuff, and so people have high blood pressure because things aren't working correctly. People have low blood pressure because things aren't working correctly, and we need to know the normal ways that things work so we know what medications and which ways we can stimulate different body parts to help aid in allowing the kidneys to function in the correct way. So just lastly, I always want to remind you that all these things, the urinary system is not in isolation from any other system. So you have to have all these systems working together in order for a human body to function correctly. So let me just highlight a couple of them. So how is the urinary system and the integumentary system, how are they related to one another? Well, the integumentary system, it does produce precursors of vitamin D that the kidneys will further refine and make into calcitriol, which is the active form of vitamin D. So they work together to maintain, the calcium homeostasis. So calcitriol made by the kidneys is going to tell the digestive system to increase its intake of calcium. So the lymphatic system, the urinary system produces an acidic urine, which helps to rid the body of pathogens coming up the urinary tract. I guess you could go on and on and on, but without vitamin D, the muscular system and nervous systems won't work because vitamin D helps to absorb calcium, and that's critical for their functioning. Of course, without calcium, you can't have strong skeletal system, and of course, your nerves won't work, and your muscle tissue and your heart won't work without the absorption of calcium. The respiratory system, because it gets rid of hydrogen ions and reabsorbs bicarbonate, is actively helping to maintain pH in addition to the respiratory system's activity. Okay, well, we could go on and on about the interrelationship between the two. All right, well, I thank you for your attention. I hope you learned a little bit about the urinary system, and I hope that you will study, definitely read, text material, and go back and look at sections that may be confusing to you. So next time we'll study a different topic, and until next time, I will see you then.